My name is David Silver, and I'm a professor at Duke NUS in Singapore, where I serve as the Deputy Director of the program in cardiovascular metabolic disorders. And my research interests focus on understanding very fundamental biology and questions in physiology regarding how cells and tissues store and transport lipids. So it's commonly appreciated by both doctors, the public, the lay public, that DHA is important for brain growth and development. The brain does not synthesize DHA. This is an important fact. And so there must be a way to transport that in from the blood to the brain. And that's one of the major contributions of our research, is to discover the biochemical mechanism, identifying Mfsd2a as that transporter at the blood-brain barrier. Moreover, we discovered that a specialized lipid produced by the liver is a carrier, that's called the LPC. The protein is open to only one part of the cell at any given time. So pointing up would be outside the cell. So it engages with its molecule, in this case LPC. It binds to it, undergoes a conformational change, and flips to the inside, facing the inside of the brain, and releases its content, which would be DHA-LPC. And all of these proteins, these MFS proteins, tend to work by the same mechanisms. Now, if you go to the health food store and you buy DHA, that would be in the form of a triglyceride molecule. So that's a fat. And largely that's harvested either from microorganisms or from fish liver. However, the brain does not transport that chemical form. It has to be converted into a lysophospholipid, or an LPC, by the liver. The liver is responsible for converting your dietary DHA intake into an LPC DHA. That LPC is the molecule that the brain sees through Mfsd2a. So what are the sources of LPCs that you could find in nature? And one is in Arctic krill, which naturally synthesizes phospholipids containing DHA. Another might be in fish eggs. What is really very interesting and surprising to us is in our discoveries we found several families with mutations in Mfsd2a. And those families that have these mutations, the children in those families that have these mutations, present with very severe underdeveloped brain, or microcephaly, very similar to the rodent models that we generated in the laboratory. This is the first human genetic evidence linking brain development, cognition with DHA. That's quite remarkable, and that's very compelling evidence that LPCs are critical for human brain growth and development. That's the only genetic evidence, to my knowledge, linking that. Based on a lot of clinical studies that we know, preterm babies have various issues with cognitive development. DHA is associated, or we should say correlated, with cognitive development and healthy outcomes. So if you put all of that clinical data together, the discovery that LPC-DHA is the major form of DHA that's delivered to the brain, we would anticipate better outcomes in babies that are treated very, very early on. These are preterm babies, rather than wait or give another form of DHA which needs to be processed by the liver.